Hi everyone. Today we are going to continue with where we stopped from chapter 3. So today we are going to talk about the cytoplasm and some of its organelles. All cellular material is located between the plasma membrane and the nucleus, and it's composed of the cytosol, the inclusions, and the organelles. We can divide the cytoplasmic organelles into two types, two categories, which are membranous and non-membranous. The membranous category, the organelles, they're confined and restricted by a membrane, which is composed from the plasma membrane, and the non-membranous ones, which does not have a membrane. So under the membranous category, we would encounter the mitochondria, endoplasmic reticulum, Golgi apparatus, peroxisomes, and lysosomes. The non-membranous category, you will encounter the ribosomes, the centrioles, and as well as the cytoskeleton. The importance of membranous organelles would be like membranes, it allows compartmentalization, which is crucial for the cell functioning. And let's look at some of the organelles. First, let's look at the mitochondria. Mitochondria, it's called as the power plant of the cells because it's produced most of its cells' energy molecules in the form of ATP, and it produced through the aerobic respiration, which is the respiration which requires oxygen. And the mitochondria, it composed of two membranes, so we call it as a double membranous structure. And the inner membrane, it has many folds, and we know that when you have many folds, you increase the surface area. And these folds, we call them as cristae. The cristae, they play a role in the cellular respiration. And mitochondria, they do contain DNA. So that's a key important thing which you need to remember, because there's only two organelles which has DNA, which is the nucleus as well as the mitochondria. And mitochondria does have RNA as well as it has some attached ribosomes as well. So with related to the ribosomes, they are membranous organelles, and it's the site of protein synthesis. Ribosomes, they are made up of protein, and the type of RNA that you encounter in ribosome is the ribosomal RNA. There are three types of RNA, mRNA, messenger RNA, rRNA, ribosomal RNA, and then the transfer RNA, which is tRNA. So the type of RNA you can find in ribosomes would be rRNA. For ribosomes, there are two switchable forms, which is like some would find freely at the cell itself, cytosol, and sometimes it's being membrane bound. So like for RAFIR, that the ribosomes are bound to the endoplasmic reticulum, and that's why we call it as RAFIR as well. So that is what you need to remember too. The endoplasmic reticulum, it consists of series of parallel interconnected cisterns, and the cisterns are flattened membrane tubes which are very closely stacked with each other, and their interior is being filled with fluid. So the other feature would be that they're continuous with the outer nuclear membrane, and there are two types of ER, which is the rough ER and the smooth ER. So if you look at this figure, you would see this is the nucleus, and the outer membrane of the nucleus is continuous with the ER, and these are the cisterns, which are the stacks, and you would see in here in the microscopic view electron that these stacks are much more closely compacted together. The rough ER is the site of synthesis of the proteins, and it's been incorporated with some more ribosomes. And also, it's a site of synthesis of many plasma membrane proteins as well as phospholipids. What will happen at the cisterns would be that the produced proteins at the attached ribosomes, inside of this ER, that these proteins are get modified as they go through these cisterns, and final protein at the end would be enclosed into vesicle, and then they sent them to the Golgi apparatus for further processing. So the smooth VR, on the other hand, it's a network of loop tubules continuous with the rough ER, and enzymes can be found in its plasma membrane, which are part of the integral proteins of its plasma membrane. And they do function as like in the lipid metabolism process, which are that smooth ER is involved in cholesterol and steroid-based hormone synthesis and making lipids from lipoproteins, and also in the process of absorption, synthesis, and transport of fats, and detoxification of certain chemicals, converting glycogen to free glucose, storage and release of calcium. So those are the functions of the smooth ER. And of course, with related to the calcium storage and release, you would encounter lots of smooth ER in the cardiac and skeletal muscles, because lots of calcium is involved with the muscle contraction as well as cardiac contractility. The other major important organelle would be the Golgi apparatus, which is a stacked and flattened membranous cistern sacs. It does function as modifying and concentrating as well as packaging proteins and lipids which received from the rough ER. In this process, there are 3 steps involved. That is, it transports vesicles from the endoplasmic reticulum, and these transport vesicles from the endoplasmic reticulum. They fuse with the cis, which is the inner side of the Golgi, and then it enters into the Golgi, and then the proteins and lipids taken inside, and they get modified, they can get tagged, they can sorted and packaged, and it's like labeling and sending out. It's like a little factory. Try to sort it out to where it needs to go. So that's why it's called tagging and sorting. And then at the end, it's going to be put out from the trans or the outer face. So the Golgi is like a traffic director, which is like controlling which of the 3 pathways that the final product needs to go. And from the trans surface, trans outer face, that these vesicles moves outside. So here in this figure, you would see that the transport vesicles comes and bind with the cis face, which is the inner face, and then that is the receiving side for the for the transport vesicles. While they go through in these systems, it goes modification, tagging, sorting, and then get back into the vesicles. These are the modified proteins and the lipids, and then they put out from the trans phase, this shipping side of the Golgi apparatus. So it's like a little factory, if you would think of. Here once again, it will show that how it's been transported from the cis surface here, that from the rough ER, the vesicles coming and merged with the cis surface, and they pass through, get modified, and then put out as packaged vesicles from the trans surface. And then sometimes these vesicles would merge with the plasma membrane and then do exocytosis is happening, like put out its secretions out of the cell. The other major important organelle would be the peroxisomes. So peroxisome, also a membranous sac, which contains powerful detoxifying substances, and it will neutralize as toxins. And especially due to the metabolism of the cell, there are lots of free radicals are being produced in our body, and if we don't neutralize it, that would harm to our body. So these free radicals, they're very toxic, they're highly reactive molecules, and they are natural byproducts of the cellular metabolism. So two main detoxifiers are present in the peroxisome. They are the oxidases and the catalases. The oxidases, they use oxygen to convert toxin to hydrogen peroxide, which is itself toxic, and the catalases, they converts the hydrogen peroxide to the harmless water. So oxidases, catalases works together. And peroxisome, of course, they play a role in breakdown and synthesis of fatty acids. If you have taken biochemistry, you would know like the involvement of the peroxisome in this process. So the next one would be the lysosomes. Lysosomes, they're also spherical membranous structure, and it contains some digestive enzymes, which are acid hydrolases. And they digest ingested bacteria, viruses, and toxins. They can degrade non-functional organelles. So it works as kind of a little scavenger. And the metabolic functions would be break down and release glycogen, break down and release calcium from the bone. And also, if the components are released while it's inside of the cell, it will release all its hydrolases, the digestive enzymes, and then the cell get destructed by itself. So we call this process as the autolysis. The clinical important aspect of some of these organelles would be with related to the lysosomal storage disease, that these lysosomal enzymes are mutated and then they do not function properly. And in Tay-Sachs disease, where the patients lack lysosomal enzymes needed to break down glycolipids in the brain cells, the glycolipids of those cells going to build up, and that would interfere with the function of the nervous system, and the patient would manifest as presenting with seizures, mental retardation, and then blindness. And these patients would usually do not survive. They would die before the age of 5. So when you think about the organelles, we would encounter a word called endomembrane system. So endomembrane system, it consists of membranous organelles, which we have discussed so far, as well as the nuclear and the plasma membrane. And these membranes and organelles, they work together to produce, degrade, store, and export biological molecules, and also to degrade potentially harmful substances within the cell. The next interesting topic that we need to discuss is the structure, like the foundation of the cell, which is the cytoskeleton. It's an elaborate network of rods which run throughout the cytosol, and it acts like a bone system, like a skeletal system or ligaments, which gives the supports to the cellular structure. And it also involved with the movement process. So with related to the cytoskeleton, we can divide its components into three types, which is microfilaments, intermediate filaments, and the microtubules. So the microtubules, they are the thinnest of all cytoskeletal elements, and they're very semi-flexible strands made up of actin protein. And each cell, they has their unique arrangement of these strands, but almost all cells share kind of a common terminal web structure. Some microfilaments involved with cell motility, some involved with changes in the cellular shape, some involved with transport mechanisms such as endocytosis and exocytosis. So here in this picture, it's been shown the different types of microfilaments, the three types of the cytoskeletal elements, which here the microfilaments, this is the intermediate filaments, and the microtubules. So here the strands made of spherical protein subunits called actin. Here you would see these actin subunits present in these microfilaments. And with related to the intermediate filaments, they're very tough. They're insoluble proteins which constructed like woven rope-like structures, and like each subunit composed of like 4 fibers. And the microtubules, they are spherical protein subunits, and the protein encounter is the tubulin. With related to the intermediate filament, it's the size in between the microfilaments and the microtubules, so that's why we call it as intermediate filaments. They are very tough, as I mentioned before, insoluble, and then rope-like protein fibers. So how it get its toughness is because it's composed of tetramers, like 4 fibers twisted together, and when you make 4 fibrous twisted together, you will get much a strong fiber itself. So it helps resist pulling forces, and some have special names which we call as neurofilaments in nerve cells and keratin filaments in epithelial cells. With related to the microtubules, they are largest of the cytoskeletal elements, and it consists of hollow tubes composed of protein subunits called tubulins. So the protein that you encounter in microtubules is the tubulin. And they are constantly, these tubulin subunits, they're constantly being assembled and disassembled. And most you encounter at the area of the centrosome. And these microtubules, they do a function of determining the overall shape of the cell and the distribution of the organelles. And many organelles, they attach the microtubules to keep the organelles in place, and many substances, they moved throughout the cell by motor proteins, and which use the microtubules as their track. So that is the important aspect. So the other important organelle would be the centrosome and the centrioles. So centrosome is located near the nucleus, and it means the cell center. And the centrioles is the pair of barrel-shaped microtubular organelles which lie at right angles to each other. And you would see here they're lying at right angle to each other. And the centrioles form the basis of the cilia and the flagella, which helps to move certain molecules on top of the cell surfaces, or even for some cells itself. So since we encountered the word cilia and flagella, let's talk about the cellular extensions. So certain cells, not all cells, have structures extending from their cell surfaces. So they would be either cilia, flagella, microvilli. And with related to the cilia and flagella, they aid in the movement of the cell or the materials across the cell surface of the cell. The microvilli, on the other hand, they are like finger-like projections which extend from the surface of the cell, and that because of its finger-like projection, it increases much more surface area of the cell. The cilia, they are much more whip-like motile extensions on the surface of the certain cells, such as you encounter them at the respiratory system, respiratory cells, of course. And the flagella, they're much more longer extensions which propel the whole cell. So one of the popular example would be the sperm, the tail of the sperm. Both structures, the similarity would be that they're made up of microtubules and synthesized by the centrioles that are called the basal bodies because they form the base of each the cilium and the flagellum. So the difference, there are slight differences though. So the cilia and flagella, they have 9 to 2 pattern of microtubules, which means like there are 9 sets of double tubes surrounding a central pair of doublets. And you would see that it in a picture in a few minutes time. And it's slightly different from the centriole because in the centriole you get the 9 plus 0 pattern where there's 9 triplets, not the doubles. In the centriole there are 9 triplets with no tubules in the center. So cilia, they moved alternatively between the power stroke and the recovery stroke. This is an alteration, this power stroke and the recovery stroke, alternative movement produce a current at the cell surface, and that would lead to movement of the substances forward. So in this figure, you would see that the 9 to 2 patterns over here, which is the doubles, 9 doubles, if you count 1, 2, 3, 4, 5, 6, 7, 8, 9, and there are only 2 units here, like doubles, and you would encounter 2 central microtubules as well. But of course, in the here you would get the triplets, but you don't get the middle 2 microtubules. So this is the 9 plus 0 pattern on the other hand. So this is what it's been shown by the power stroke, which is the propulsive stroke, and the recovery stroke when the cilium is returning to its initial position. So that will lead to movement of substances through the surface of the cell. The other important cytoskeletal element would be the microvilli, which is the little finger-like extensions of the plasma membrane. And as we said before, it increases surface area. And you would think like, which cells needs much more surface area, especially the absorption, where the organs where needs much more absorption. So definitely you would see them at the intestinal or kidney tubule cells. And it does have a core of actin microfilaments, which is used for stiffening of the projections. All right, so the next very important organelle would be the nucleus. So nucleus is the largest organelle, and it contains the genetic library of blueprints for synthesis of nearly all cellular proteins, because you would see the DNA carries all the important information which leads to formation of a protein. So most cells, they're uninucleate, which is like they have only one nucleus, but some cells, such as the skeletal muscles, certain bone cells, they do have, or sometimes the liver cells, they are multinucleated, which is like they have many nuclei. And there are some cells that does not have a nucleus, which is the red blood cells, or we call them as anucleate. The nucleus has 3 main structures: the nuclear envelope, nucleoli, chromatin. The nuclear envelope, it is a double membrane barrier which closes the jelly-like fluid, which is the nucleoplasm of the nucleus. And the nuclear membrane or the nuclear envelope, it does contain nuclear pores, which allows substance to go in and out. and it does not allow freely to go them, but it's been guarded by the nuclear pore complex, which regulates the transport of specific large molecules. And inside of the nucleus, there's an area called nucleoli, which is a dark staining spherical bodies, and it's involved with the ribosomal RNA synthesis and assembly of the ribosomal subunits. So the questions can come as like where the ribosomal subunits are going to be assembled. So you need to know that is the nucleoli. So nucleoli is associated with the nuclear organizer regions, which contain the DNA that codes for the ribosomal RNA, and usually 1 or 2 nucleoli per cell. The chromatin is also encountered in the nucleus, which is consist of 30% of thread-like strands of DNA, and 60% of chromatin is with histone proteins, and 10% would be from RNA. And so the composition of chromatin question, if they ask, you need to know that is DNA, histone proteins, and RNA. So chromatin, they arranged in fundamental units called nucleosomes, and which consist of DNA wrapped around the histone proteins. And chromosomes are the condensed chromatin. So further condensation of nucleosomes leads to formation of the chromosomes. And this condensed state helps to protect the chromatin being get broken down during the cell division process. So that is the very important feature of the condensation. So here it will show us that the chromatin and the chromosome structure, you would see like this is a bigger magnification, and you would see like these are the histone proteins, the purple color ones are the DNA. So the DNA is wrapped around, if this is the histone protein, it's wrapped around this histone protein, the DNA, and then they further get twisted and condensed, and you can see how many nucleosomes involved with formation of even a small length of a DNA chromosome segment. So that is actually the end of the chapter 3 part C, which is the second component, and we will start the latter component, the last part, which is the cells and its living units, which we need to talk about the cell cycle and the mitosis process. So let's talk about the cell cycle. So if you think about cells of your body, some cells, they continuously dividing. Some cells, once they differentiate, they can decide like, I'm not dividing again. So cell cycle is involved with the reproduction process, but you would see like the differentiated and non-dividing cells at the cell cycle too. So like all cell, like the human being, we have particular lifespan. All cells, they do have a lifespan. So during that lifespan process of the cell, they do need to reproduce and make another cell. And especially we would see with the process of tissue repair process, the cells are dividing. So let's go ahead and look at what's this all about and what it does need to have to do with the cell cycle. So cell cycle, if we have to define, it's a series of changes a cell which undergoes from time it's formed until it's reproduced. So as you can see in here, it can be divided into 2 major phases. So look at this arrow pointing where it's going to, the direction as well. So starting from here, G1, all the way at the end up to G2 is the interphase. And during this phase, the cells grows and carries on its usual activity. So it's in order. It's the growth 1 phase, S phase, and the G2 phase, growth 2 phase. Sometimes in textbooks you will encounter these as gap 1 phase, but that is fine too. So G, growth or gap 1 phase. The S stands for synthesis. So what does it need to synthesize? The DNA. So the DNA is synthesized in the interphase, especially at the synthesis phase or the S phase. Then at the end of the growth 2 phase, what is happening is the mitotic phase. And if you look at this figure, you would see the mitotic phase can further divided into two major things, mitosis and the cytokinesis. So the mitosis, I would ask the students to remember as PMAT, which is the prophase, metaphase, anaphase, and telophase. And this is very easy to remember if you understand the very basics. So the subphases of interphase would be the gap 1 phase, growth 1 phase. In the growth 1 phase, there are vigorous growth and metabolism happening within the cells. Some cells, they're permanently cease dividing, and during the gap 1 phase, those are called like the G0 phase. And so those cells, rather than going with the gap 1 phase, they go into G0 phase. The synthetic phase where the DNA replication happens, and the gap 2 phase where the cells prepare for the division. It's not yet started, it's preparing. That you need to remember. With related to the DNA replication, we need to know certain important aspects. Prior to cell division, cells needs to make a copy of their DNA to give it to its daughter cell. So, of course, we know that DNA is a double-stranded helical structure. It has to unwind because it's been twisted and winded together. To make a copy, you have to open up those two double strands, and therefore you have to unwind it or unzip it. And the point where the strand starts to separate is called the replication fork. And the active area of the replication where the happening is, we call it as the replication bubble. And each strand during the replication process acts as a template for a new complementary strand. On the other hand, RNA, they starts replication by laying down short strands, which acts as a primer. So the enzyme involved with DNA replication is the DNA polymerase. What it does is it attaches the primer and begins adding nucleotides to form the new strand. So the primers are RNA, short segments of RNA strands. The DNA polymerase, they synthesize both new strands at one time. That is, you can label one strand as the leading strand and the other one as the lagging strand. The leading strand, it's going to be produced continuously. The lagging strands, it will happen like forming little segments in places, and then at the end it's going to be ligated at the end. So that's why it's called the lagging strand. But the leading strand, it forms continuously. So DNA polymerase, it works only in one direction. So leading strand is synthesized continuously, but the lagging strand is backwards. It's synthesized discontinuously into segments, and at the end, the DNA ligase, it splices short segments of the discontinuous lagging strand, splices, which means like edit, and short segments of the discontinuous lagging strands combined together. So the end results would be you're forming two identical daughter DNA molecules which are formed from the original strand. And during the mitotic cell division, one complete copy will be given to a new cell while one is going to retain in the original cell. So therefore, we call this DNA replication as the semi-conservative replication, because each new double strand of DNA is having one old strand and one new strand. So let's look at this in a picture. So here you would see that this is the leading strand. Of course, you would see that it's been continuously formed over here, and the lagging strand is formed as segments, and then at the end it would be formed as a complete strand by the help of DNA ligase. And of course, with the unwinded one, you would see like this strand is from the old template strand and these strands would be from the new strand. So that is why it's called the semi-conservative replication. With related to the cell division, the mitotic phase of the cell phase is phase in which cells division is happening, and you can divide that into two major ones, which is the mitosis and the cytokinesis. So during the M phase, which is the mitosis, is the division of the nucleus, where the duplicated DNA is going to distribute it to new daughter cells. So as I mentioned before, there are 4 stages: prophase, metaphase, anaphase, and telophase. And you need to remember at least 1 or 2 key features happening at these phases. The prophase, it can further divide into early prophase and late prophase. So early prophase, before you start the nuclear division, the chromosomes, they are very dispersed and they're loosely, it's very loose. It's like a strand, it's a coil strand has been loosened up. So it's all over the nucleus. So the very first thing happening is chromatin starts to condense. And when they start to condense, they start to form visible chromosomes. So that is a key feature happening at the early prophase. Condensation of chromatin and visibility of the chromosomes. And each chromosomes now is being duplicated because it has formed another new strand, because it has to give it to a new daughter cell. So we use the term sister chromatid for a duplicated chromosome. So that is important. And these duplicated chromosomes, they're held by a center, we call it as the centromere. So these centrosomes and its duplicate begin synthesizing microtubules, and they push each centrosomes to opposite poles of the cell. So don't get confused. Centromere is a different one from the centrosome. So you have to, if you don't remember what is centrosome, you need to go back into the beginning of the lecture and look what is a centrosome is. So at the beginning, the centrosomes, they're also duplicated. They start to synthesize microtubules. They're going to go to opposite poles of the cell. So imagine thinking like the cell is like the world, and you have the north pole and the south pole. The duplicated centrosomes is like a satellite, 2 satellites, and one satellite is going to go to the north pole, the one go to the south pole. While they're going to those 2 poles, they're making like microtubules. So we call these microtubules, which span from the one pole to the other pole of the cell, as the mitotic spindle. And the other microtubules, called the asters, they can radiate from the centrosome. So this is what it's been mentioned. So here is the asters which radiate from the centrosome. They are much more shorter. And these are the microtubules, which are the microtubule spindle from the centrosomes. And you would see now here the chromosome starts to condense, and you would see a little bit of little linings, and you would see like the nuclear membrane is still intact. In the late prophase, what will happen is the nuclear membrane breaks up. That is a key feature to look for. So you don't see a nuclear membrane over here, but you see much more condensed chromosomes, and the special microtubules, they attach to specific areas of the centrosome, which are called the kinetochore, and they serve to pull chromosomes to the center of the cell. And the remaining non-kinetochore microtubules, they pushed against each other and they cause poles of the cell to move further apart. So here, if you look at this, you would see there's two centrosomes over here, the other centrosomes over here. You see the kinetochore where the chromosomes have been attached to, and you would see the microtubule, spindle microtubule fibers. And during the metaphase, it's very easy to remember. That is, like all the chromosomes, they lined up at the equator line or the center of the microtubule spindle. So if you look at the cell, you would expect to see a single line which arranged at the center of the microtubule spindle in the metaphase. And we call this plate, this is an imaginary plate, we called it as the metaphase plate. During the anaphase, it's the separation of the sister chromatids. So you would see this pole, it will pull up, the microtubules get shortened, it pulls up the chromosome. So the chromosome separates, the sister chromatid, they separates from the centromere. And because of the pull, it acquire kind of a V-shaped structure. So this would go to this side pole, and these chromosomes would go into the other side of the other pole. And at the end of the telophase and the cytokinesis, at the end of the telophase, actually the cytokinesis started to happen, which is for laying out the cell membrane in between these two cells. So, and as sister chromatids, they have already gone to both sides. The new daughter cells start to form from the cytoskeleton, which is the cleavage furrow, which we tell. So the control of the cell division is important aspect as well. It's like go and stop signals which direct when a cell should and should not divide, because the go signals include the critical surface to volume ratio of the cell when an area of the membrane becomes inadequate for exchange. Then you would ask like more cells to be produced, and they are the ones for growth factors. The go signals would be the growth factors, the hormones. The stop signals when the availability of the cell's space is going to reduce, then the normal cells needs to stop dividing, and that would happen if a cell is divided and then you occupy the remaining space. So you don't have more space to further division. So the cell-to-cell contact would give the signal to stop. Okay, now it's enough. Let's stop the cell division like that. So this is referred to as the contact inhibition. Two groups of proteins are involved with the cell division process, which is ability to accomplish whether the S phase and the entire mitosis. These are the cyclins and cyclin-dependent kinases. So cyclins are the regulatory proteins which accumulate during the interphase, and the cyclin-dependent kinases, in short form, we call them as CDKs. They activate cyclins when they bind to them. So the cyclin and the CDK, they form a complex, and they in turn activate enzyme. We call it cascades, like when there are several enzymes regulated by another enzyme. For instance, if you have A, B, C, D like that, the A enzyme is needed to work to function the B enzyme, and then the B enzyme activation is needed to function for the activation of the C enzyme. So it goes like a hierarchy and a regulatory process. So we call them as a cascade. And the cyclins, they are destroyed after mitotic cell division, and the process begins again. They do not stay forever. Like once their task is done, the degradation happen of that cyclins. And also, when a cell is divided, there are several checkpoints there because there can be lots of errors can happen. So checkpoints are key events in the cell cycle where cell division process are checked, and if faults error or if errors happened, then what they do is they stop the cell division until that error is be corrected or repaired. So there are several checkpoints. So these are the ones at which is at G1 checkpoint, which is the near end of the G1, which is closer to the G1 and the synthesis S phase transition. And G2 checkpoint is near the end of the G2. And the spindle checkpoint is partway through M phase and more specifically at the metaphase or the anaphase transition point. Because if there is an error and if it's not corrected, there's no point of further cell division. And sometimes with related to certain cancers, you can escape this checkpoint. So that is the other fascinating aspect as well, that there are mechanisms which leads to escape of these checkpoints as well. So G1 checkpoint, we call it as a restriction point, is the most important of three major checkpoints. So if the cell does not pass, it enters into G0, and which is no further division is going to happen with those cells. With related to the nucleus and its function and what is happening inside of the nucleus, we need to discuss about the protein synthesis. So DNA is the master blueprint which holds a code for the protein synthesis, because if you think about, we have structural proteins, we have functional proteins, and almost the living being is functions with these protein functions. So the DNA is the one which keeps the information and decides which protein is going to be produced. So a segment of DNA which holds the code for one polypeptide is referred to as a gene. And I hope you would remember what is a polypeptide means, because lots of amino acids combined and form a one polypeptide. And we learned about its primary structure, secondary structure, tertiary structure, and the quaternary structure as well. So this is just like a one polypeptide. So the code is determined by the specific order of the nitrogen bases, which is the adenine, guanine, thymine, and cytosine in the gene. And the code consists of three sequential bases, which we call them as the triplet codes. For examples, if you have guanine, and cytosine, it codes for amino acid proline. But if you have guanine, cytosine, it codes for the arginine. So that is like several patterns of this code gives rise to different amino acids. And each triplet specifies the code for a particular amino acid. That is the other feature. There are, with related to the protein synthesis, we need to know the other important aspect would be the exons and the introns. So genes, they're composed of exons and introns. Exons are the part of the gene which actually codes for amino acids, and the introns are the non-coding segments which interspersed between the exons. So these are nice MCQ points or true false statements. So you have to know that exons are the ones which codes for amino acids and introns are the ones which does not. With related to the RNA, so RNA plays a role with related to its function involved with the protein synthesis. So RNA, ribonucleic acid, is the one which go between the molecules that links the DNA and the protein. So that's why we need to discuss that in here. So RNA, they copies the DNA code in the nucleus, and the ribosomes are at the cytoplasm. So it has to take the message which is created at the nucleus into the cytoplasm and has to translate it into a protein. So all RNAs is formed in the nucleus. That is the other important aspect. And if you specifically remember, rRNA at the nucleoli. So RNA differs from DNA from its one type of amino acid, which is the uracil, or is substituted for thymine in RNA. So in DNA, you will find thymine. Instead of thymine, in RNA, you will find uracil. So also RNA, it does have ribose sugar instead of deoxyribose sugar. So that's why we call it as DNA, deoxyribose nucleic acid. And RNA is just the ribose nucleic acid. So that's the major two differences. And there are structural differences as well. DNA double-stranded, RNA single-stranded as such. But basically, you need to remember the very important, at least these two. So three types of RNA is present in the cell, which are the messenger RNA. And if you look at this little lowercase letter, it implicates what it stands for. So messenger M stands for messenger, mRNA is for messenger RNA. The R stands for ribosomal RNA, and then the T stands for transfer RNA. So messenger RNA, it's a single strand and it codes from DNA template strand. It code from DNA template strand, and it copies the DNA template strand, formed complementary bases for the DNA strand, and that will result in an mRNA. And this process, we call it as the transcription. And mRNA maintains its triplet code, and when you have three nucleotides, three nitrogen bases together, we call it as a triplet code, or we call them as a codon, and they maintain this triplet code from the DNA. The ribosomal RNA, on the other hand, is a structural component of the ribosomes, where the organelles, where the protein synthesis occurs. The ribosome is the site of protein synthesis. And along with tRNA, the ribosomal RNA helps to translate the message from mRNA into a polypeptide. We call them as the translation process. The role of RNAs, the transfer RNA, the tRNA carries the amino acids. And if you look at this structure, at this 3' end, 3' end actually, we call it as the 3' end, attached with an amino acid. And you have the anticodon, which complementary to the codon of the mRNA. So here you have the mRNA strand, which is from 5' direction to the 3'. And if you look, if you make this little bended lineage structure, which is a tRNA, into one line, you will encounter 5' tally with the 3' end, and 3' end tally with the 5' end. They are very complementary, anti-complementary here. So here the codon GCC, you would see it's the complementary nitrogen base would be this G for C, and G for C again for C for C. So, and with related to the transfer RNA, they carry of amino acid. They have special areas which contains a specific triplet code, which is the anticodon, and it allows each tRNA to carry only a specific amino acid. And the anticodon of tRNA is complementary base pair with the codon of the mRNA at the ribosome, and adding its specific amino acids to the growing polypeptide. So we call this as the translation process. So with related to the protein synthesis, there's two steps involved, that is the transcription and the translation. So transcription is where the DNA information is coded into an mRNA molecule. So there's a little writing error, like M has to be lowercase, RNA has to be uppercase. The translation is where the mRNA decoded to assemble polypeptides. So transcription is happening at the nucleus, inside the nucleus, where the DNA, from the DNA, an mRNA molecule is made, and then the mRNA molecule travels across the nuclear post into the cytosol, and the translation now is happening at the cytosol. So because the ribosomes are here, and then the ribosome, they formed a polypeptide chain. That is why it's called as the mRNA decoded to assemble the polypeptides. With related to the transcription, it's the process of transferring code held in DNA gene base sequence to the complementary base sequence of the mRNA. So lots of transcription factors important. These transcription factors are protein complexes, and these transcription factors are needed to activate the transcription process. So the transcription is broken down into three phases. That is initiation, elongation, and the termination. So let's look at each one of them, at least in a few ways. So initiation is where the RNA polymerase separates the DNA strands, and elongation happens when the RNA polymerase, they adds the complementary nucleotides to the growing mRNA, matching the sequence of the base sequences based on the DNA template strand. Because unlike the DNA replication process, if you have a strand of 10 micrometers, that the DNA replication process, it happens throughout the 10 micrometers. But in here with the transcription process, not all genes are transcribed at one time. Certain genes get transcribed, certain genes get untranscribed. So the genes get transcribed at particular moment. The DNA strand is now twisted at that time, so it has to unwind like in the DNA replication process. So at the initiation process, the RNA polymerase is the enzyme which involves with the separation of this DNA strands. And now you have to create an mRNA strand from a DNA strand. So what will happen is the same enzyme, the DNA polymerase, they adds the complementary nucleotide into the RNA, into complementary nucleotides, and that will create a mRNA molecule, and this will grow to the needed length. And the short 12 base pair segments where DNA and mRNA are temporarily bonded is referred to as now the DNA-RNA hybrid. And at the end, once it's finished, when it's needed to finish its function, the transcription stops when the RNA polymerase reaches the special terminated signal code, because for transcription to happen, there is initiation code as well, and once that initiation happens, elongation is happening, that is like growing off the mRNA strand, and you have a stop signal to say, okay, that's enough, this is where you need to stop. So that is the termination signal, where the termination happens. So here what it's been shown so far, what we have discussed. So with the initiation, with the help of transcription factors, the RNA polymerase binds to the promoter region. This is the RNA polymerase. It's going to bind to the particular DNA promoter region of that gene, and then it's open up the DNA strand, and you start to make the mRNA strand to that relevant DNA strand. And once with the elongation process, this is the initiation, you will make that mRNA strand longer as the polymerase moves along the DNA strand. And with related to the termination, once it's encountered the termination signal, which is shown in here, that it will stop its mRNA synthesis, and now the mRNA strand, which is the completed mRNA strand, passed from the nuclear pores into the cytoplasm. Before it's passed into the cytoplasm, at the inside the nucleus, the mRNA is going some changes, which is the processing of mRNA. So the newly formed mRNA is then edited and they processed before translation can begin. So what will happen is, we call the unprocessed mRNA is as the pre-mRNA. What will happen is the introns, remember, those are the regions which does not produce any protein. So those are being removed, and we call that process is called spliceosomes and splicing. And the proteins involved the splicing process is the spliceosomes. And after splicing, you leave only the exons, coding regions of the mRNA. This processed mRNA is being transported into the cytosol of the cell, and the second step starts to begin, which is the translation process. So steps of the protein synthesis, translation is where the second step, and it's a step of the protein synthesis where the language of the nucleic acids is now translated into another language of proteins, which we called as the amino acid sequence. So if you think about the translating, which is like, you know, one language and you translate into somebody else's language. So it's the same thing, like you made a DNA strand. Now you have to make an amino acid strand. So therefore, you have to translate that DNA message into an amino acid sequence. So the process involves the mRNA, the genetic code, the tRNA and ribosomes, the translating events, and sometimes the rough ER. So lots of things involved with the translation. So let's look at the genetic code first. So each 3 bases sequence of the DNA is represented by a complementary 3 base sequence on the mRNA called, we call it as the codon. So there are 64 possible codons, which is like how we got the 64 is we have 4 bases and the codon does have only 3 places. So 4 into 3 means like 64. 4 into 4 is 16. 16 into 4 is 64. So that's how we got the possibilities of 64. There are 3 stop codons, but rest are codons for amino acids. So out of 63, only 61 will code for amino acids. And also there are only 20 possible amino acids, and these 20 possible amino acids combined or they assemble in different ways, and that's why you get different types of polypeptides. So if you look at, there are 64 codons. Ideally there should be 64 amino acids, but there's a now reduction, 20, only 20 amino acids, but there are now 64 codons, which means like some codons, maybe 2 or 3 codons, can code for one type of amino acid. So this process we call as the redundancy. So the redundancy helps to protect against some transcription errors. So therefore, that's why it's been important with related to the protein synthesis. So here in this genetic code, you would see what I said. So here you have one codon, another type of a codon, but all of these 4 codons, they code for one amino acid, which is the serine. And some would be like only 2 codons for aspartic acid, like that. So therefore, the redundancy, even though there are 64, there's only 20 amino acids would be coded from this genetic code. And the ribosomes, they coordinate coupling of mRNA and the tRNA. And ribosomes, it contains 1 binding site for mRNA and 3 binding sites for tRNA. So the amino acyl site for amino acyl tRNA, those are the 3 binding sites. The amino acyl site, we call it as A site in short form. The peptidyl site, we call it as the P site. The peptidyl site is for the tRNA linked to the growing polypeptide chain, and the exit site, the E site, for the outgoing tRNA. And like the transcription process, the translation also happening at 3 steps: the initiation, elongation, and termination. And it requires protein factors, energy currency, ATP, as well as enzymes. So here it shows in the picture like the initiation process, elongation, and termination. So let's go and look at one at a time. So with the initiation process, it's occur when there's 4 components combined at the P site. Here you would see the P site over here, and this is the E site, this is the A site. So what will happen is 4 components would be a small ribosomal subunit, because the ribosomal has a small subunit and a big subunit, the larger subunit. So the smaller, a small ribosomal subunit is involved, and an initiation tRNA has to carry the amino acid methionine to initiate, and you need to have an mRNA molecule and a large ribosomal subunit. So these 2 subunits has to combine, the larger one and the smaller one, and the larger one has to be there, the mRNA molecule has to be there, and the transfer RNA has to be there. So those are the 4 things at the initiation place. The elongation is happening is when the amino acids are added one at a time to the growing polypeptide. And you see here, this is just the first amino acid, and then you will see like a bead kind of a structure. This is the polypeptide, and each time with the elongation process, that the amino acids is going to be added. So you would see that it's in from 2A to 2B, that this elongation process. So in 2A, it shows the codon recognition, the anticodon of an incoming tRNA. This is the anticodon part. They bind to the mRNA molecule, complementary mRNA codon in the A site of the ribosomal mRNA codon in the A site of the ribosome, and then the peptide bond is formed between each other, and then what will happen is the tRNA at the P site is now transferred to the amino acid carrying the tRNA in the A site, and then a new peptide bond is formed between these two. The translocation process is happening. All include the codon recognition, peptide bond formation. Translocation comes under elongation process. So in the translocation process, the entire ribosome then translocates, shifting the position one codon along the mRNA. So it shifts like one to the one forward. So therefore, whichever at the P site is now go to E site and the next set, and then it's move forward A site, you get a new codon, and for that one a new tRNA molecule brings up. So the termination process is when you encounter a stop codon. There are three stop codons, UGA, UAA, UAG. When the arrives at the A site, then the elongation starts to end. So this is what it shows, like this is UUA, UGA actually. So you would encounter a stop codon, and then you don't start proceeding. You don't proceed. So what is the role of the rough ER in the protein synthesis? So that is important as well, because ER is one of the components we seen involved with the protein synthesis process. So a short amino acid segment called ER signal sequence present in the growing polypeptide chain, and this will signals the associated ribosomes to dock onto the rough ER surface, because it will, that little signal in the polypeptide chain carries it to the ribosome and carried to the endoplasmic reticulum, and that will dock into it. And the signal recognition particles, we call them as SRP on the ER, they would direct the mRNA ribosome complex where to dock. And once they docked, the forming polypeptide, they enter into the endoplasmic reticulum. The sugar groups would be added into the protein, and once they incorporated new things, the shapes would be altered of that protein. It could become a glycoprotein, sugar and protein, or sometimes lipid components get added into the protein. So we call them as the glycolipids. Not lipoprotein, sorry, lipoprotein. So like that, several things, several importance, mainly sugar and protein would be added into these proteins. And these proteins then can be enclosed in vesicles, and they transported into the Golgi apparatus. So here you would see what will happen with related to the SRPs. They direct the mRNA ribosomal complex to the rough ER, and then the SRP binds to the receptor site of the rough ER, and once they get attached the ER to the ER, the SRP is going to release, and the growing polypeptide then get into the ER membrane, get into the ER through the ER membrane pores, and then the enzymes involved with clipping off this signal sequence, and then the sugar components are going to be added into this polypeptide, which is the glycoprotein now, and they were packed into vesicles, and they are now transported into the Golgi apparatus. So all in all, why we learn this is like how these things going to be applied into clinically. So we learned about cell cycle. We learned a lot with related to this chapter 3. Lots of transport mechanisms, lots of organelles, and when they're gone wrong, what will happen, and so on. So cells, with related to the cell division and the things that we have discussed with this part, cells can get damaged, and sometimes if it's a permanent damage, it needs to be taken out of the system. So there's process called autophagy, which is like self-eating. In this process of disposing of non-functional organelles and cytoplasmic bits by forming autophagosomes, which can then be degraded by the lysosomes. And sometimes the unneeded proteins can be marked for destructions by a protein called ubiquitins. How they do is the proteasomes, that disassembles the ubiquitin tracked proteins and recycling the amino acids, and the ubiquitin is happening at that process too. With related to the apoptosis, which is, it's a programmed cell death, because each and every cell has a lifespan. At the end, it has to get rid of from the system itself. So it's such as the self-destruct, and this apoptosis can happen in cancer cells, infected cells, or old cells, whatnot. And the process begins within the mitochondrial membranes, because mitochondria are composed of lots of caspases. These membranes, when the apoptosis process happens, the from the mitochondrial membranes, the leaking chemicals can lead to activate of the enzymes called caspases. And these caspases, they can cause degradation of the DNA and the cytoskeleton, and eventually the cell starts to die, and then the dead cells is going to be taken up by the phagocytic cells of the immune system, which are the macrophages. And with related to the cell aging, because we have talked about the apoptosis and autophagy, as the humans, because humans are made from cells, of course, they do get old. So the mechanism of aging is still not kind of clear, but several theories have been put out. Three theories is put out so far: the wear and tear theory, which is a lifetime of chemical which insults the free radicals and it has a cumulative effect. The mitochondrial theory of aging, where it says like the free radicals in the mitochondria, it diminishes the energy production. And then the immune system disorders such as the autoimmune response, as well as the progressive weakening of the immune response, would lead to the aging. And also another type would be the genetic theory, which is kind of the acceptable one, which is the cessation of the mitosis and cell aging are programmed into genes. So in each one of our chromosomes, we have a portion called telomeres, and they're strings of nucleotides which protect the ends of the chromosomes. It's like caps or shoestrings. So each time when a cell divides, the telomeres get shortened. So it may act like an hourglass on how many times a cell can divide. So more the telomeres get shortened, then the lifespan of that cell is going to be reduced. So there are telomerase existing, which is an enzyme that lengthens the telomeres. And it's been said that it's been found in germ cells of the embryos, but absent in adult cells. Except this is present at cancer cells. So that's why the cancer cells' existence of telomerase will lead to survival of these cells throughout. Okay, so this is the end of chapter 3. You definitely would need to read the chapter to get more comprehension. And I will meet you at the next class. Thank you.